Hello dear students, welcome to the next online lecture of your DNA structure. In the previous video we discussed about the basic concept related to the composition of the DNA. What are the types of composite that are present to make a particular double helix DNA. So the major composition of DNA is belonging to the three criteria. First one is your nitrogen bases, second one is your ribose sugar, deoxyribose sugar, that is nothing but the pentose sugar, and third one is like your phosphoric acid or the phosphate group. After that we studied about the purines. Like the nitrogen bases are divided into the two types, one is purine and another one is pyrimidines. In that, purines also divided into the two groups, one is adenine, second one is guanine, and pyrimidines having the three types, cytosine, thymine and uracil. Instead of thymine, uracil is present in the RNA. So next one is that the bonding. DNA is a double helical structure and that is held by, अगर आपका डीएनए जो मॉलिक्यूल है वो डीएनए हेलिकल है, आपने कभी लैडर देखा होगा, डीएनए भी एक लैडर की तरह है लेकिन वो हेलिकल लैडर है, ठीक है। वो हेलिकल लैडर जैसे हम सीढ़ी चढ़ते हैं तो वहाँ पे जो दो डायरेक्शंस होते हैं पैरेलल होते हैं और उसमें सीढ़ी के लिए हमें एक स्टेप बाय स्टेप ऊपर जाना होता है, ठीक है। उसी प्रकार जो सीढ़ी है वो जो मेन कंपोजिट है उसका उससे हेल्ड किया जाता है, ठीक है। वैसे ही मतलब लैडर, और वैसे ही डीएनए हेलिकल का जो स्ट्रक्चर है इट इज आल्सो हेल्ड बाय द नाइट्रोजन बेसेस एंड इन दैट हाइड्रोजन बॉन्ड प्लेज अ वाइटल रोल. Okay, so let's see the interactions or the bonds that involve in the DNA structure. So the double DNA helix is held together by the two types of bonds. One is covalent and the hydrogen. Covalent bond occurs within each linear strand and which is the strongly bond with bases, sugar and phosphate groups. Hydrogen bonds linkage between the bases, although a weak energy wise, it is able to stabilize the helix because of the large number of molecule which is present in the DNA. Also there is a, here you can see that this ladder of double DNA helix, it is held by the hydrogen bonding, okay, and it is the main composite to hold the DNA double helical structure, okay. The next stacking interaction is base stacking. The bases, which bases? The DNA bases like nitrogen bases in which adenine which pairs with the thymine with double hydrogen bond, guanine which pairs with the cytosine with triple hydrogen bond. The base stacking interaction are also known as the van der Waals interaction between the bases, ठीक है. So what actually I want to explain you that stacked base, मतलब यहाँ पे आप देख सकते हो ये base stacked है, ठीक है, that attract to one another through the van der Waals forces or van der Waals interactions, okay. Major stacking forces are hydrophobic interactions and van der Waals forces, okay. So next one is the phosphodiester bond. Phosphodiester bond, here we can see that where is the phosphodiester bond is present. Yes, the phosphodiester bond is the covalent backbone of the DNA which is present or which is linked formed between the 3' carbon of the one molecule and that link to the another 5' carbon of the sugar of deoxyribose, okay. So here you can see that the 3' carbon atom which is linked to the 5' carbon atom of the deoxyribose sugar in the DNA, and the phosphodiester bond is the strongest bond or the covalent bond which is present in the DNA. So this is about the bonds involved in the DNA structure, okay. Then the last topic is the structural forms of DNA. In RNA, there are types of RNA like messenger RNA, rRNA, ribosomal RNA, tRNA, transfer RNA. These are the types of RNA. But the DNA don't have any types. They have the structural forms. Okay, structural form means they are having the different types of structure which is present in the DNA molecule, and that is present in the vice versa or the different types of species. Okay, so different forms of DNA. Although we all know the finding of Watson and Crick नाम के जो साइंटिस्ट थे, उन्होंने एक्सरे क्रिस्टलोग्राफी से बी फॉर्म ऑफ डीएनए इसका स्टडी किया था. ठीक है, सो बेस्ट स्टडीड मॉलिक्यूल जो है वो बी फॉर्म है. सो बी फॉर्म ऑफ डीएनए मेनली देयर आर 3 टाइप्स ऑफ डीएनए, और सॉरी फॉर दैट, स्ट्रक्चर नॉट द टाइप्स. ओके, structural forms of DNA, they are A form of DNA, B form of DNA and Z form of DNA. Rather than this, C form DNA, D form DNA and E form DNA is also there. Okay, but the major forms of DNA are A DNA, B DNA and Z DNA. So let's see one by one what are the different characteristics of particular form of DNA. So bDNA is the Watson-Crick form of the double helix. 2 strands of DNA, each in right hand helix. So helix handness, here you can see that aDNA having right helix, bDNA is also having the right helix, but the zDNA, it is radically different duplex structure with the 2 strands coiling in left-handed helix and pronounced as a zigzag DNA. Okay, which is having the zigzag pattern of phosphodiester backbone. Okay, the base pair turn, aDNA is having the 11 base pair turn, bDNA is having the 10.4 base pair turns, and Z form is having the 12 base pair per turns. Okay, ZDNA is actually having the aDNA is actually having the 10.5 to 6 base pair turns, but it is okay. So approximately it is 11. Okay, some of the books are saying 11, some of the books are saying 10.5. So we can refer the standard book. Okay, rise per base. So what do you mean by the rise per base? I am explaining in the next image. So we will see that rise per base is of aDNA is 0.23 nanometer, bDNA is 0.34 nanometer and zDNA is 0.38 nanometer. Pitch is nothing but the differences between the bases that align to the particular helix molecule. So aDNA is having 2.46 nanometer, bDNA is having 3.14 nanometer and zDNA is having 4.56 nanometer. Diameter of the aDNA is 2.55 nanometer, bDNA is having the 2.37 nanometer, zDNA is having the 8.84 nanometer. Conformation of glycosidic bond which is present in the sugar and the nitrogen bases. aDNA is having the anti-syn conformation, bDNA is having the anti-conformation and the zDNA is having anti and syn conformation. Major groove, minor groove, these are the grooves where we can see that in the next slide, the figure. aDNA's major groove is present, bDNA is present, zDNA may absent because zDNA having the zigzag pattern of conformation. So this is the basic differences between the different forms of DNA, and that helps to understand the varieties of the forms, structural forms of DNA. Let's see the figure one by one. Here you can see that the particular structure of the B form of DNA, in that the diameter of the double DNA helix is 20 angstrom. Here you can see that the minor groove is present, the major groove is also present. Helix pitch is 33.2 angstrom of the B form of DNA. Okay, so next one is your axis rise or axial rise is 3.32 angstrom. So this is about the particular structure of B form of DNA, in which the two long polynucleotide strands coil around the central axis. Strands are wrapped plectonymically. Plectonymically means right-handed, or sorry, in the form of ladder. Plectonymically means ladder shape. Okay, and it is in the right-handed helix. Strands are anti-parallel. One strands that orient from 5' to 3' direction, and another strand is oriented from 3' to 5' directions, and strands that interacted by the hydrogen bonds which is present between the complementarity base pairs. G forms 3 hydrogen bonds with C and A forms 2 hydrogen bonds with T. So this is a particular structure of your B form of DNA. In that all the concepts are now clear, that diameter, minor groove, major groove, helix pitch, the differences we have seen in the previous slide. Now it is well explained. So Z form of DNA is the left-handed helix and it is the zigzag. So here you can see that particular A form of DNA, B form of DNA and the Z form of DNA, it is zigzag in shape. Okay, because of that it is named as zigzag DNA or the Z DNA. The major differences in between these three forms of DNA is that A form is right-handed helix, B form is a right-handed helix, but only the Z form of DNA is left-handed origin or the orientation. Okay, and there is also difference of base per turn. 10.7 base per turn it is present in the A form of DNA, 10.4 base per turn which is present in the B form of DNA, and Z form of DNA 12 base per turns are present. Okay, so this is about the different forms of DNA. Rather than this, I also said that C form of DNA is there, D form of DNA is there, and E form of DNA is there. Also, other than these forms of DNA, there is a triplex DNA. It is present when there is a circumstances like low pH or the DNA sequence containing a long segment of polypurine strand. Okay, the extra strand added in the DNA helix. So triplex DNA. And also, other than this triplex DNA, there is a G quadruplex DNA which is rich in guanine. That's why it is known as G quadruplex. These have the 4 standard structures of guanine. So it is also known as G quadruplex or G quartet. Okay, so with this we finish about the structure of DNA and the concept related to the DNA molecules. Okay, thank you all of you. And if you have any questions, so you can ask in the comment section. Other than, you can also ask in the SYBSC microbiology group. Till then, go through the structure of DNA and basic concept related to the DNA, because that will help you to understand the next lecture of DNA replication. Okay, thank you.